Can you please turn off the lights? All right. Thank you very much to all of you for being here. I'm Imtiaz Ahmad. I'm postdoctoral researcher in the Department of Entomology, working with Dr. Mary Babachek. So in a previous, since a couple of years, we have been trying to understand the biology and ecology of insect pathogenic fungi in organic cropping systems. And we have also been trying to understand how different crop management practices affect the abundance and prevalence of these insect pathogenic fungi. So today I'll be talking about some of our research from those projects. Plants in the environment, they they face many challenges in the environment, and to cope with such challenges, plants have evolved multi-mechanistic strategies. One of the strategies that plants have evolved is their interaction with the microbes. And there are many factors that influence this interaction, that influence this association. For example, biotic and abiotic stresses and climate conditions, microbial community that can affect the plant directly, or the plant microbe interactions, as well as crop management practices, rhizome microbiome, and soil properties. So these are the factors, some of the factors that influence the plant microbes interactions directly or indirectly. So we need to consider that microbes are very important part of our ecosystem, and we need to conserve that. We need to build a healthy community of the microbes in the soil. So because of so many microbes present in the soil, we think that our soil is as alive as we are. So these microbes have there are different microbial communities by nature, as well as due to their diversity in their nature, they are also diverse in their functionality. And a healthy agricultural microbial soil can contain millions of microbes in it. Those microbes vary in their diversity, genetic diversity, as well as their functional diversity. In the environment, plants interact with different biotic factors or organisms, pathogens, and beneficial microbes, insects, nematodes, oomycetes above ground, and as well as below ground. But our focus is specifically the beneficial microbes and beneficial fungi in this category. We are trying to understand a group of insect pathogenic fungi that are soil-borne. They live in the soil, and they have been widely studied. And you can see these yellow dots show where these insect pathogenic fungi, specifically I'm talking about Metarhizium, that has been studied. Different species of Metarhizium has been studied around the globe. So this fungus, this group of fungi is ubiquitous, has been found almost everywhere in specifically agricultural environment, in agroecosystem. So this is common multifunctional soil inhabitant that can establish mutualistic plant association as well as parasitic association with the insects. So two-pronged benefits, benefit to the plants by plant growth promotion, as well as benefit to the plant by insect pest suppression. So these are also has been reported as pathogens of different phytopathogens. So there are many layers of the benefits that these fungi harbor for our agroecosystem. And it has been reported that these are very well adapted to agroecosystem. And we think that this adaptation to agroecosystem may be due to their ability to be dispersed due to the disturbance that happens in agroecosystem. So more disturbance, more management practices in our soil can lead to the dispersal of the spores that are present in the soil, and better dispersal may result in better in better association of these insect pathogenic fungi to find a new host, insect host, as well as plant host, because we have from previous literature we have seen that there are many Metarhizium species that have been found in association with different plant types. So plant type definitely depends, as well as the species of the Metarhizium that they interact with. Metarhizium completes its life cycle in 3 different associations. And those associations are saprophytic life cycle, when Metarhizium is present in the soil and its spores are fungal material feed on organic matter as a source of their survival nutrients. And that is one of the source of further life cycles for Metarhizium. And from there, in the soil, they can form association with insect hosts, susceptible insect host, as well as they can form association with the plants in rhizosphere, as well as endophyte, plant endophytes. In their interaction with insects, there has a lot been studies going on related to Metarhizium and insect relationship, and has been widely studied, and we can see that this infection starts from from spores that are in the soil. Those spores, when present in the soil, come into the contact to the body of the insect. They differentiate to the infection structures. They differentiate into the infection structure, and through mechanical penetration, as well as by the help of different enzyme lipases and hydrolytic enzymes, they degrade the cuticle, penetrate the cuticle of the insects, and after penetration, they propagate into the body of the insect to form further hyphal bodies. And after penetration, they cause intoxication, feed on the nutrient contents of the insect, and after feeding and after finishing up all the nutrients in the insect, the insect becomes killed, that becomes cadaver, and from the cadaver, this fungi come out by producing more spores. So few spores can produce millions of spores, and that can start a new infection cycle to a new insect host. This is about their pathogenicity in the insects. So this is one part of their association. The other association that plants undergo is with plant. The Metarhizium undergoes is with the plants, and in plants, as I said, it has been reported that Metarhizium has been found as rhizosphere, because rhizosphere is an ecological hotspot, specifically in the rhizosphere, and there are studies that these are also endophytes of the plants. Some researchers still believe that this is not the endophytes and this is only rhizospheric interactions, but there are many other studies that have established the fact that this is predominantly, dominantly the root endophyte. And when present in association with the roots, and when present in association with the insect, first they kill the insect, as I have shown you the mechanism, how it happens. And then after being associated with the insect, what they do through these fungal hyphal network, it has been studied that it has been reported that they translocate nitrogen from killed insect to the plant. So this was really very important study, because for other fungi, arbuscular mycorrhizal fungi and different other fungi, beneficial fungi, it has been reported and that they are nutrient, they help in nutrient transfer and nutrient translocation. But for this specific group of fungi, it was never reported before. In 2012 and 2017, the same group suggested that not only this, because this is mutualistic association. So what is this fungus getting from the plants in addition to habitat? Is this just the habitat? No, it's not just the habitat. It is also feeding on the sugar contents of the plants. So what is from this trade-off, what it is getting is the sugar. So translocating nitrogen from killed insect in the soil to the plants, helping the plants to grow better and protect them, as well as in return getting sugar as a source of carbon. So recently in 2017, there has been a study published that has shown that Metarhizium in association with plants can also help the plant root, development of the plant root architecture, help in lateral root growth and secondary roots in Arabidopsis. And this has happened through the auxin-dependent mechanism. So phytohormones play a vital role. There was first study published in 2012 that gave some evidence, but there was still lack of evidence that phytohormone, that this fungus can still mediate the phytohormones in plant growth and defense. So in 2017, this study showed that Metarhizium is involved in root architecture growth and development through auxin-dependent mechanisms. So phytohormones play very important role in this, as it is evident for different other beneficial microbes as well, because microbe-mediated phytohormone production and defense in plants is very well evident. And there are different phytohormones that are involved, for example, auxin, cytokinin, brassicinosteroids, gibberellin in plant growth promotion, and in plant defense, ethylene, salicylic acid, abscisic acid, and jasmonic acid. Our focus is specifically on jasmonic acid and salicylic acid. Excuse me. Jasmonic acid and salicylic acid, and jasmonic acid specifically being, you can see in the center, although this is very important, all the phytohormones are very important for different defense and growth related functionalities, but jasmonic acid is really one of the primary pathway that is involved in plant defense and growth. So jasmonic acid, depending upon the organism that plant is being associated, for example, plant is being associated with is interacting with biotrophic phytopathogen or biotrophic microbe or necrotrophic microbe, these pathways differ. So jasmonic acid is involved in defense against chewing insect pests and necrotrophic and symbiotic fungi. And this also acts as a switch between growth and defense with other phytohormones. Whereas salicylic acid is involved in plant defense against biotrophic phytopathogens and phloem-feeding insects. And for many plant species, it has been reported that both of these pathways work antagonistically for plant defense and growth. So when plant is present in the environment, it's the ideal condition would be it is not facing any threats, but that is not true. It's always some threat to the plant. So plants are always facing those challenges. And for example, theoretically, if we say if there is no stress, then plants need to fine tune their plant growth defense pathways. And to do that, to fine tune that, what plants do is they switch on and switch off certain pathways, because mounting a plant defense is not free. Nothing is free. There is cost associated to that, energy cost. So for that cost, what plants have evolved is their strategy to switch on and switch off certain pathways just to balance. But in the presence of stress, what it could do is, for example, it could mount up or it could switch on defense pathway and could switch off different pathways related to growth. There is a third model that suggests that not only this, plants are so efficient that they can also balance both of these, growth and defense. So there is integrated model that says that plant can grow by switching on, switching off certain pathways related to growth and defense in a balanced way. So what we wanted to understand from these perspective is the ability of Metarhizium robertsii that we have, that is very dominant in our research at our research station. The ability of this Metarhizium robertsii to form endophytic relationship with maize in greenhouse conditions. And if there is endophytic relationship, then how does it affect the maize growth? How does this association affect the biotic stress, the black cutworm, and key defense genes of the plants that are involved in plant growth and defense? So what we do is we collect soil from our agricultural research station, and we bait that soil with waxworms. And after baiting for a few days, if there are spores in the soil, then Metarhizium spores can infect the susceptible Galeria melanella waxworms. And then we get a pure culture. We store these and do the molecular identification by using translation elongation factors. It's very interesting that through this method of molecular identification, from all of our research sites, whether that was research station, whether that was commercial farm, we collected sample, and there was only one species of Metarhizium present. That was Metarhizium robertsii. And maybe that is the dominant, maybe that is more competitive related to other species of Metarhizium in this area. We have sampled different farms in Pennsylvania, as well as New York, nearby farms, 8 farms. So there were around 130 samples that we did molecular identification, and we only found one species. So we, excuse me. Yeah.
Is the wax worm only one particular genotype that you're using to capture the?
They are found that this is reported that they are susceptible to Metarhizium. Being susceptible, I mean, there are different genotypes, but usually there is no difference in the defense because they don't have very good defense mechanism for this, for insect pathogenic fungi. That's why it is used as a sensitive, because it's sensitive and we can detect Metarhizium proportion in the soil. That's why it is used.
But I guess my question is it also sensitive to all the other
insect pathogens?
Microbiome.
Like
robertsii and all the others.
It is susceptible to insect pathogens, insect pathogenic fungi. So we have found in our experiments Bavaria bassiana, that is not Metarhizium, that is another group of insect pathogenic fungi. So it has been found that this is not the only fungus that it can infect. When we do evaluation, we categorize in our soil sample whatever we got, whether that was Metarhizium robertsii, whether that was Metarhizium Bavaria bassiana or Aisaria. So different insect pathogenic fungi can infect these worms. So we soak the seeds of maize for 2 hours, and after soaking, we grew the plants in the greenhouse for till V4 stage, and then we evaluated root and leaf tissue for endophytic colonization. We evaluated plant growth and plant defense and black cutworm growth. In our feed experiments, what we have found in organic rotation of maize soy wheat, we have found that Metarhizium is more present in maize phase of rotation. So that was very interesting as compared to soy and wheat. So we wanted to understand this biology as well. So we conducted experiments in controlled condition because in ecological perspective, it's very difficult to dissect the mechanism and dissect the true factors that are involved. So we wanted to understand why it prefers Metarhizium, what is the endophytic colonization in maize, so how does it colonize? So in our experiments, we found 91% of endophytic colonization in root and leaf, and that was mostly the systemic. So you can see we categorized the plants into 3 categories, whether that endophytic colonization was only in the leaf tissue, whether that was only in the root tissue, or that was both leaf and root tissues. So we found that Metarhizium robertsii can establish as endophytic colonization systemically in 91% of plants. And when we further evaluated for leaf and root endophytic colonization, we found that there was higher proportion of root colonization as compared to leaf colonization. So root may be like many other microbes, beneficial microbes, root is definitely one of the favorable habitat. Maybe this is also the case for Metarhizium robertsii, that or the other reason we think is maybe this was the first point of contact with the spores leading to higher endophytic colonization over time as compared to leaf tissue.
Excuse me, Imtiaz, can you clarify that in your previous slide? When you say leaf and root,
yeah,
you said, is that the addition of
all of those?
Leaf and root?
Yeah.
So you're combining
leaf and root tissues.
Okay, so that's basically the addition of the 2 bars on the right side.
Yeah, okay. Where we have found, like we categorize the plants into 3 categories, whether that was only found in leaf tissue, whether that was only found in the root tissues, and this is total, if we combine that, total is 91%.
So let me see if I can, so the bar on the right, leaf and root,
yeah,
is a plant where you found Metarhizium in both the leaf and the root.
Yeah.
A single plant with
yeah,
colonization in both places.
Yeah.
And do you have any idea of whether that is actually the same fungus in both places, or if they could be 2 different? Can Metarhizium colonize from leaf?
Yeah.
It can.
In
So
our experiments, we did, yeah.
They could be two different fungi.
Could be, could be, but what we did is in our control plants, if we think that this Metarhizium is coming from aerial infection, we don't think, because Metarhizium in control plant, we didn't find any Metarhizium sample in control plants in the same environment. So that means they are all coming from the root inoculation and going above ground. So this is not something coming aerial infection.
Could you explain again how you see them, the endophytic fungi? Like, is it culture-based? How do you reveal?
Yeah, so you can see these small pictures. So these are root tissues. What we do is we surface sterilize the root tissues, and we plate onto potato dextrose agar medium. And after 2 weeks, this fungus is growing and sporulating from those plant tissues. So that's how we evaluate whether the plant tissue or whether the plant was colonized or not. And this is culture-based method. Yeah.
So you're working with the single strain?
Yeah, yeah. Just single strain.
Test any different, if there was any different strains?
For some of the isolates we studied in our experiment, because there were many isolates that were isolated. For some of those, we did the molecular identification. We selected some from the root tissue and some from the leaf tissue, and they all were the same, that the one that we inoculated. Yeah, we confirmed that through molecular identification. Did you get your answer? Okay. So we measured the plant height, and we then categorized the plants into 3 categories: not inoculated, control plants, the plants that were inoculated and from which we detected Metarhizium robertsii, and plants that were inoculated and we could not detect Metarhizium robertsii. So we found that Metarhizium inoculation increased plant height. Increased plant height in inoculated and detected plants as compared to control plants, and this colonization was positively correlated with tissue colonization and phytocolonization. So we think that this may be due to the higher and the greater endophytic colonization might have resulted in better root architecture that might have helped plants to absorb better nutrients as compared to control plants. Similarly, we also measured plant biomass, above ground biomass, and we found that the inoculated plants, as was the case with the plant height, had higher, greater above ground biomass as compared to control plants, but this effect was only correlated with root colonization and not with leaf colonization. So we think that this may be the same mechanism that have helped plants to grow better, to grow higher, and similarly might have contributed to better above ground biomass. We measured chlorophyll content of the 4th true leaf of maize plants, and we did not find any significant difference in control versus treated plants. But that effect was positively correlated with leaf and root colonization, although that was not significant. The difference in the chlorophyll content was not significant, but there was positive correlation. So that was the physiological part of the plants. Then we studied what's going on at molecular level, what's going on at chemical signature level. So because beneficial fungi, beneficial microbes can trigger plant immunity, can help plants grow better by protecting them, by providing defense through molecular signatures, microbe-associated molecular patterns. So we evaluated the plants for Jasmonic acid and salicylic acid pathway. And in Jasmonic acid and salicylic acid pathway, we studied first the Jasmonic acid biosynthesis pathway. So we studied lipoxygenases. Lipoxygenases are involved in diverse array of different plant growth and defense mechanism. And these are involved in biosynthesis of precursors of Jasmonic acid. Similarly, we also studied OPR7, that is oxophytodienate reductase 7. There are also a group of enzymes that are involved in the biosynthesis of precursors of Jasmonic acid. So we wanted to understand what's going on in the upstream in this pathway. So we found that the plants that were treated with Metarhizium robertsii, that were grown from treated seeds, had upregulation of lipoxygenase 1 and OPR7. And we think that this may be due to the plant perceiving Metarhizium robertsii as a biotrophic phytopathogen, and that might have elicited the accumulation of plant defense compound and the precursors of Jasmonic acid that were upstream in this pathway. Similarly, we also wanted to understand what's going on downstream. What's going on, that was biosynthesis pathway. What's going on in the Jasmonic acid and salicylic acid response pathway? Is plant responding to that if the Metarhizium is phytopathogen or not? So we have seen that plants have showed response in biosynthesis, but in case of response pathway, we studied maize protease inhibitor. That is a marker of herbivory in plants. And because there was no stress to the plant, because there was no insect feeding, no herbivory going on, that's why we think that plants might have not responded to the treatment of Metarhizium robertsii. And you can see there is downregulation in the treated plants of maize protease inhibitor. We also studied pathogenesis-related protein 5. That is the marker of the pathogen defense in the plants. So this is one of the precursors compound that is produced, that plants produce, the antifungal compounds in response to pathogens. So we have seen that Metarhizium robertsii treated plants had upregulation of salicylic acid, pathogenesis-related protein 5. And we think that this may be due to the plant perceiving Metarhizium robertsii as biotrophic phytopathogen and eliciting salicylic acid pathway. And salicylic acid pathway is also, here you can see, that is also evident for many other plant species, that it acts antagonistically to jasmonic acid. And there is very fine line, like plants are very efficient in determining who is their friend, who is their foe. They can distinguish between those by using molecular and chemical signatures. So that's why I think plants might be sometimes able to or not able to distinguish between phytopathogen or beneficial fungi that are in interaction with the plants. One of the other strategies that plants have evolved is the biosynthesis of different proteins, toxic proteins. These are chitinases. The chitinases degrade chitin-related compounds. We have studied endochitinase A and as well as pathogenesis-related protein 4. They both are endokitinases. The endochitinase A is involved in defense against biotrophic phytopathogen, whereas pathogenesis-related protein 4 is involved in defense against necrotrophic phytopathogen. So we think that because Metarhizium robertsii is not a necrotrophic phytopathogen, that might have resulted in downregulation of pathogenesis-related protein 4, whereas upregulation of endochitinase A.
Imtiaz, can you clarify, is this leaf tissue or root tissue?
This is leaf tissue, only leaf tissue. In our future experiments, we are planning to do both, like root, and we are also trying to understand what happens if the insect is a root herbivore. So this was done with because that was leaf herbivore, above ground herbivore. So that's why we focused on above ground parts.
And that's black cutworm?
Yeah, that's black cutworm.
Okay, thanks.
We studied the relative growth rate of black cutworm because black cutworm is one of the herbivore that is early season insect pest of corn and causes huge economic loss, cuts the plant just above the ground. So we try to understand what happens because our plants were not too old. They were up to 21 days after germination, that is V4 stage. So we wanted to, and we conducted this assay with detached leaf. We used detached leaf. We fed the black cutworm, second instar black cutworm, with leaf tissue for 96 hours and studied the relative growth rate. And we found that the growth rate of black cutworm larvae that were feeding untreated plants was lower as compared to control plants. And this is also the data from, if you remember the data from biosynthesis pathway, Jasmonic acid biosynthesis pathway, that was upregulated, the genes were upregulated, that's showing that there may be the accumulation of certain defense compounds in the plant tissues. So that might have resulted in maybe less feeding of the insect, or maybe less growth or development of the insect, resulting in lower growth rate in treated plants. And this effect was negatively correlated with leaf and root colonization. So because of its insect pathogen application as well as plant growth promoter, Metarhizium has been developed into commercial products, but these commercial products are available only for ornamental plants or vegetable production, not for field crops, because it's not viable to use at this point to use the Metarhizium treatments. It's available in liquid treatments as well as in granular treatments. With this, I would like to summarize my talk that Metarhizium.
Can I ask a question?
Yeah, sure.
Back to the comment you just made. So is there the potential for using them for field crops? What would it take to, would you have to reduce the cost of the production of the Metarhizium? Would it have to be more effective in the?
Yeah, I think it's more related to mass production of Metarhizium, because in field crops, it's not economical. You need a lot of seed treatment, maybe not volume, but you need a lot of volume of seeds to be treated. So I think it's that may be one of the reasons that it's not being developed for field crops.
How long does it persist in the field? Can you apply once and then it will?
There are studies that have shown that it can persist in the field up to 3 years. I have seen a study that was conducted for 3 years. They inoculated the field and then evaluated yearly, although the level decreased in that specific plot. Although it's very difficult to study in natural conditions, but it can persist for 3 years for sure. But I think it varies because there are many other factors, soil properties, as well as temperature, as well as humidity. So there are many other factors that lead to its persistence in the soil. And from my experiments that I was doing in the field, I'm not showing the data for that right now. We have found that Metarhizium robertsii does not look like a good competitor of other microbes in the rhizosphere, or because we have found very less endophytic colonization. So that might be because we use surface sterilized seeds, although there was a lot of stuff going on in the field. But it's we try to understand if that is something we could understand, if Metarhizium can infect in the field and can grow as endophyte. But as endophyte, that might be helping the plant growth promotion through root exudate formation or by changing the chemistry of root exudates. But endophytically, we did not find a lot of Metarhizium robertsii in the field, in the maize. So in our experiments, we recovered Metarhizium robertsii from 91% of the plants, and none of the control plant was infected. We found systemic recovery mostly, and endophytic plants had better plant growth, above ground biomass, and altered plant defense gene expression in Jasmonic acid, salicylic acid pathway, as well as the expression level of plant chitinases, plant-derived chitinases. Relative growth rate of black cutworm was lower when fed on plants that were treated with Metarhizium, that were endophytically colonized with Metarhizium. And these data suggest and support the integrated model of plant growth versus defense response in response to stresses. So we are further trying to understand different mechanisms related to this. We are further escalating this research to understand how this Metarhizium robertsii affects the Cochleobolus heterotrophus. Cochleobolus heterotrophus is one of the causative agents of southern corn leaf blight. That also causes huge economic loss around the globe. We are trying to understand, because there are some reports related to Metarhizium robertsii that this is involved in phytopathogen defense in plants. So we are trying to understand what happens with Cochleobolus heterotrophus. We are also trying to understand the soil disturbance, because we are using gradients of tillage, and we are trying to understand how that soil tillage disturbance affects the natural prevalence of Metarhizium robertsii. And we will also try to connect the dots with the field, because we will conduct some experiments in the field with the soil that we will collect from the field. We will do experiments in the greenhouse as well. We are also trying to understand the effects of biotic and abiotic stresses on Metarhizium maize symbiosis. So we, in this specific experiment, we will be using combinational stress. We will be using a combination of both biotic and abiotic stresses to see if these stresses drive the endophytic colonization of Metarhizium in controlled condition or not. So if these are the factors, or vice versa, or the endophytic colonization helps plants in alleviating these biotic and abiotic stresses. So with this, I would like to acknowledge our funding sources: USDA, NIFA, Northeast IPM, Penn State College of Agricultural Sciences, and also our project team. And thanks to all of you for your attention. Yeah.
So, if I understand your terms, you started the talk framing it in terms of Metarhizium being potentially this nitrogen transfer from a killed insect
For the
to
plant.
plant.
Mm-hmm.
But your experiments are completely independent
Yeah.
of that.
Nutrients.
So
Yeah.
you're suggesting there are these mutualistic benefits that go above and beyond and are separate from any nitrogen transfer from an insect.
Yeah, in different experiments, because that experiment was conducted with specific reference to killed insect. And we did not use killed insect as a source of phytopathogen, Metarhizium. So we think that the plant growth response, although those nutrients were not translocated from killed insect, we think that plant growth promotion effects might be due to better root architecture that might have helped in better nutrient acquisition by the plants. So although these nutrients are not directly, I mean, that was just one of the aspects showing like Metarhizium is involved in nutrient translocation, because that study was recently published, and that was really groundbreaking to show that Metarhizium is really translocating through radioisotopic study. They have shown that really killed insect, and then using those helping plants to grow better, using those nutrient resources to grow better. I think that was very interesting, although we are not doing those specific experiments, although we are trying to conduct similar experiments in our system to see uh, if if there is any fungal hyphal networking involved in plant Metarhizium association, because in trees, with the trees, so there are reports that fungi provide a network, and through those networks, fungi can help neighboring plants that have not undergone any stress communicate the signals. So we are trying to understand if this also happens in the case of maize.
Yeah, and I absolutely didn't mean that as a negative. I thought it was really interesting.
Yeah, no.
That you're basically isolating out and still getting benefits.
Yeah, I understand that. Yeah.
Can I make a comment? I think that initial report of nitrogen being transferred to corn from an insect was a gee whiz, kind of a one-off report. I mean, it was very exciting.
Yeah.
But I would, want to point out that that probably doesn't happen very often, but it
Yeah.
was shown directly with stabilized terms.
Yeah.
So it probably doesn't happen.
Yeah, probably does not happen. And I said there is scarcity of data related to plant, Metarhizium, and defense. There is a lot of research going on related to Metarhizium and insects, but this is specific field really needs attention.
They were characterizing the relationship as making plants carnivores.
Yeah.
Yeah.
I was wondering if you looked at the below ground biomass at all, and you see that it's possible that there's increased nutrient acquisition, but you also said that there were increased pathogenic gene expression. So could it be that the is actually reallocating resources
Yeah.
above ground?
You are right. We did not do the experiment. We did not conducted the evaluation in the below ground, but that is really very interesting and something we definitely are focusing and planning to do in the future experiments, that we should definitely use below ground herbivores as well as we want to study what's going on at the root level. Initially, what the reason we didn't do the below ground biomass, because it's really very difficult to get a reliable data from the roots. I mean, while washing, you can still waste a lot of biomass. So that was one of the reasons that we thought it would probably not give us good results, but we are planning on doing that.
So you showed that Metarhizium is using some defense mechanism.
Yeah.
Do you know there is effect?
Transgenerational in the plant. We don't know about studies that have shown the transgenerational effects, but we know that plants remember this, because I hesitate to say that there is priming effect going on in the plants, because priming is a memory that transfers to the generation. So because our experiments to confirm that this was priming effect, we need to conduct a study with stress, in the presence of stress. That is our future goal. But right now, I cannot confirm that there is transgenerational effect in maize. We are right now, we are trying to understand two different genetic lines of maize and to see what happens in this association in that case. Then probably I'll be able to answer that.
I have a question about your isolation of the very same Metarhizium robertsii based on the
Yeah.
colonization factor sequence. So you probably know that several of us have got a paper out in a publication in Phytobiomes. So what we did was we did bulk soil microbiome analysis. This was kind of a post hoc study that was based on a visiting scientist who wanted to learn some molecular techniques. So we did analysis of bulk soils in about, 8 different cover crop treatments, and this was during cover crop growth, and then after cover crop harvest, and then planting of the corn. So we did, and it was based on fungal ITS. It was not based on the elongation factor.
Yeah.
And so we did detect Metarhizium and Nisopleae, and several other. We haven't talked to you about this.
Yeah, we did, yeah.
So we have detected just based on DNA, and it could be relic DNA or extracellular DNA. But I'm wondering if there's something about the way you're collecting the dominant Metarhizium from your larvae, because I think you're collecting it from after larval infection, and it just might be really, really competitive.
Yeah.
Even within the larvae. So I'm just pointing out there might be diversity.
We talked about it, and we thought that why not we do some other molecular method instead of baiting. We tried to, so the isolates that we isolated from plant tissues that were naturally grown, we tried to we did molecular identification of those as well to see if plant is picking up something different than what insect was picking up, but we still found Metarhizium robertsii. The other thing is Metarhizium anacephali. Before 2009, that's what Metarhizium robertsii was also in the same lineage. Bishop et al. In 2009, they changed the lineage. They suggested Metarhizium robertsii belongs to Metarhizium robertsii is belongs to a lineage that consists of 9 other Metarhizium species. So, and then differentiated. Before 2009, Metarhizium anacephali and Metarhizium robertsii were considered the same thing.
Okay.
Yeah.
Okay.
So that may be one of the reasons.
That's a problem that I think Carol relates to.
taxonomy keeps changing. Yeah.
Well, thank you very much for presenting to us. Oh, I'm sorry.
It's a very short.
Sure, no problem. I think we have time.
I was wondering if once it's endophytic, if it's still connected to some IV free living, or if it relies completely on the plant for
Yeah, we don't think that it is still connected to hyphae in the soil when being endophyte. We think that there is probably the same similar mechanism that the way it infects the insect, that it goes inside the tissue. So we don't think that this is something going on that is still connected. Maybe, but we don't know about it. But we cannot say like it is.
Okay, now we'll thank you.
Thank you.
Happy Valentine's Day, everyone. Or